Okay, let's now look at metabolism of carbohydrates. How are the carbohydrates metabolized? Now, you must first know what you mean by metabolism. Metabolism basically is, when you talk about metabolism, metabolism is a chemical reaction that occur in living cells, okay? Now we have two types of metabolism. We have anabolism, and also B, we have catabolism. Now anabolism is the build-up process which require energy to form complex molecules, whereas catabolism is the breakdown of the complex molecules to form now the simple molecules, and the reaction is normally exothermic, that is, heat is released. Now I want us to look at glycolysis. Now glycolysis is a metabolic process which occurs in a living cell. Now glycolysis occurs after digestion has taken place. Now when the carbohydrate food is eaten and digested, the final product is normally called the glucose. Now glucose is taken now to the cells, okay, where it is used to form energy. Now for us, we want to see how is this energy formed. A living cell, a single living cell requires about 8 ATPs for it to function normally. Now glycolysis normally forms only 2 ATPs. Only 2 ATPs are formed in glycolysis. Now glycolysis has two phases. The first phase of glycolysis is called preparatory phase. Now this is a 5-step reaction. Then second phase is called the payoff phase. Again, this is a 5-step reaction. So in a total, we have 10 steps for the formation of the ATPs, that's the 2 ATPs through glycolysis pathway. Now I want us to look at the first phase of glycolysis. As we had said, it is called the preparatory phase. And it has 5 steps. Now in the first step, the glucose molecule is normally phosphorylated. So we have glucose, it is phosphorylated to form glucose 6-phosphate. Okay. Now the enzyme involved in this reaction, let me just call it E, is called exokinase. E is for enzyme, exokinase enzyme. Now because there is phosphorylation, we are going to add ATP. That is adenosine triphosphate. So it is like we are adding a phosphate group, okay, in this particular glucose molecule to form glucose 6-phosphate. Now, glucose 6-phosphate means that the phosphate group is added to carbon number 6 of the glucose molecule. Let me just show you here a bit so you can get to understand what we are talking about. This is the glucose molecule. This is a glucose molecule. Let's use the alpha. We are adding, remember we had said that this is carbon number 1, carbon number 2, 3, 4, 5, 6. So we are adding a phosphate group, okay? a phosphate group. We add it at this point. This phosphate group is already here there. When we do so, we form a glucose 6-phosphate. The reaction is called phosphorylation. The enzyme of 6-phosphate. So it is hydrogen and just get this one first. Now here is where we form now our phosphate, glucose 6-phosphate. So you can see how we attach our phosphate group. Okay, so this one is called glucose 6-phosphate. This is the one we have here. Now, when we remove a phosphate from the ATP, that's adenosine triphosphate, we are going to have only 2 phosphates, which we call ADP. That is adenosine diphosphate, because there are only now remaining 2 phosphate group. Now, in the second step of glycolysis, in the preparatory phase, is where we have isomerization. The reaction is reversible. So we isomerate, okay, the glucose 6-phosphate. We remove the phosphate from 6-glucose 6-phosphate and take it to another molecule which we call the fructose 6-phosphate. So there is a change of the structure of the sugar, fructose 6-phosphate. Okay? The enzyme involved is called isomerase enzyme. Isomerase enzyme. So we are interchanging the phosphate between a glucose sugar and a fructose sugar. Again, you can indicate it there. The fructose molecule remember we had said it's carbon number 6. So it's carbon number 6, 1, 2, 3, 4, 5, 6. So our phosphate will be there. Our phosphate will be there. Okay? So we call this one fructose 6-phosphate. This is the one we form here. Now, in the next step, we are going to form a compound called fructose 1,6 bisphosphate. We call it bisphosphate or biphosphate. Meaning we are going to add another phosphate molecule. That is, we add ATP there, ADP, to form. So it means that in this particular molecule, we are going to add another phosphate. Okay, we added carbon number 1 now. That's why you call it carbon number. This one means there are 2 phosphates, one at carbon number 1, another one at carbon number 6. Okay, so at carbon number 1, we're going to have this phosphate here. Okay, so you call it biphosphate or bisphosphate. The enzyme involved is phosphofructokinase. Okay, PFK, yeah? Fructokinase enzyme. So the enzyme involved in the formation of fructose 1,6-bisphosphate is called phosphofructokinase enzyme. Let's take number 3. In step number 4, in this step is where we are going to form a compound. Our molecule is going to undergo a cleavage, and the enzyme we're going to use is called aldolase enzyme. Aldolase is going to be a cleavage. Now what I mean by a cleavage, two molecules are going to be formed. Okay? Two molecules that if you're going to form, okay, at this point we're going to form dihydroxyacetone phosphate, 3-phosphate. And also you're going to form glyceraldehyde, 3-phosphate. Okay, phosphate. Remember, our fructose 1,6-bisphosphate is a 6-carbon molecule. When we cleave it, there's a cleavage here. They will say we have a cleavage. When we cleave it, we are going to form a 3-carbon sugar molecules. One is called dihydroxyacetone 3-phosphate, and the other one is called glyceraldehyde 3-phosphate. Now let's continue step number 5 and see what we are forming. Now this one cannot be hydrolyzed or cannot take part in the reaction unless we convert it back to a glyceraldehyde 3-phosphate. This is a glyceraldehyde because you have said it's a 3-carbon sugar. It has 3 carbon atoms. Okay, this is what we mean by by a glyceraldehyde 3-phosphate. Okay. We said it is 3-phosphate, meaning the phosphate is in carbon number 3. 1, 2, 3. So we have the phosphate attached there. Phosphate attached there. Now, if it was acetone, acetone is a ketose. This is an aldose sugar. This is an aldose sugar called glyceraldehyde. And then also we have a ketose sugar called dihydroxyacetone phosphate. Okay, dihydroxyacetone. For our case, it is also a 3-carbon sugar because we have said that we are breaking the 6 carbon sugar to form 3 each. Okay, so we are going to form that. Also remember again, it is the phosphate in carbon. The phosphate is in carbon number 3. It's in carbon number 3 that way. Okay, so this is a dihydroxyacetone. Dihydroxyacetone. It is a ketose. Okay, a 3-carbon ketose sugar. This is an aldehyde, so we call it a ketose. Okay, glyceraldehyde. 3-phosphate. 3-phosphate. Now this one is a aldose. We had said before in our previous lessons, okay, that aldoses, the carbonyl carbon is in carbon number 1, whereas in the ketosis, the carbonyl carbon is in between 2 ends, carbon ends. Okay. Now let us now do the 5th reaction. Will be our last reaction in the preparatory phase. That is step number 5. Remember, we are now working with glyceraldehyde 3-phosphate. Now, this molecule this particular molecule is a 3-carbon sugar molecule, and therefore it is converted to 3. What we are going to do is to change the position of the phosphate in this glyceraldehyde. So we are going to form a compound called 3-phosphoglycerate. We call it 3-phosphor. We're just going to change the position of the phosphate, okay? 3-phosphoglycerate, okay? We're trying to break the bond, okay? To make it easier for energy formation. Now, the enzyme involved here is called dihydroxyacetone. It is called sorry for that. The enzyme involved here is called glycerate dihydrogenase. Is the enzyme involved in the formation of the 3-phosphoglycerate molecule. That is how we form the 3-phosphoglycerate sugar. That is the end of preparatory phase. Okay, so we shall continue with our next phase, which is called the payoff phase, next time. Payoff phase next time. Thank you.